where I worked on a project funded by the European Union, and then I moved to Toronto, Canada. At University of Toronto, I completed my PhD in 2014. There, I did a postdoctoral research. I was a postdoctoral researcher there for about a year. I have a professional engineering license of Ontario, Canada, and I hope to obtain one in Florida as well in the near future. So, my broader training and interests are advanced physical and chemical water and wastewater treatment processes, but I'm particularly interested in membrane processes in treating water and wastewater, and for water reclamation from impaired sources, particularly focusing on the removal of contaminants of emerging concern. I also work on membrane fouling and fate and transport of contaminants of emerging concern in environmental media. I teach courses like environmental engineering, which is an introductory course, and also environmental engineering operations and processes, chemical process control, and the graduate courses include industrial waste treatment and water treatment. Now, membrane processes do a good job in treating water and removing contaminants from water. Okay, sorry, I was just checking if everything is good. It's just a question that I'll hopefully answer. So the membrane processes do a good job in treating water and removing contaminants depending on their configuration and separation mechanisms. But I'm particularly interested in the contaminants of emerging concern because they pose risks to aquatic species and human health. The CECs, or contaminants of emerging concerns, are these various classes of chemicals that are not commonly monitored in the environment, but they have the potential to cause known or suspected adverse ecological and human health effects. Now, the conventional membranes can treat very effectively various contaminants, including the contaminants of emerging concerns, or CECs, but they have their drawbacks, like the rejection of CECs are variable, and they're very compound specific. Then one of the key challenges associated with membrane treatment is that they get dirty, or what we call fouled, which will reduce the service time and cost money. So my past and recent research have addressed these issues, and I've been investigating the removal of CECs and their interactions with water, as well as with the membrane matrix. So that we not only remove them effectively, but also continue to produce the water at a reasonable scale or volume. So in our membrane processes lab at UCF, I'm trying to develop all-rounder membranes which can address these drawbacks of associated with the membrane processes. Now, if you take a commercial membrane, you can modify it using different approaches, including incorporating nanoparticles or nanotubes, nanofibers, or you can fabricate the mixed matrix or thin film nanocomposite membranes. But how now these modified membranes are going to interact with the CECs and other contaminants that might influence their removal, as well as the membrane performance in producing water, that is something is really needed to be investigated. So we have tried to modify the commercial membranes using, for example, here in this project, using an ultrafiltration membrane, which is a low pressure membrane. So we coated the membrane with oppositely charged polyelectrolytes and immobilized silver nanoparticles within the layers, within the coating, and this resulted in improved hydrophilicity of the membrane, and surface charge, and other properties of the membrane. So if you look at the same images, this is how it looks like if you take the cross section of the membrane after you coat the membrane with the polyelectrolytes. And on this plot, you can see the permeate flux on the y-axis, and this blue line represents the unmodified membrane, and the orange line represents the modified membrane that is modified with the polyelectrolyte and silver phosphate nanoparticles. You see in this fouling stage, they both follow the same trend, but after the membrane is being cleaned, the unmodified membrane continues to decline the flux, whereas the modified membrane maintains its performance. The membrane also have become, antimicrobial. As you can see, there is less attachment of bacteria, which would have, eventually develop biofilms, leading to what we call biofouling. Now, we tested the performance of the membrane, removing emerging contaminant PFAS, or perfluoroalkyl or polyfluoroalkyl substances, which are so-called forever chemicals. So we focused on the two most commonly targeted PFAS, which is PFOS and PFOA, perfluorooctane sulfonate acid and perfluorooctanoic acid, PFOS and PFOA respectively. So because of the functionalization, the membrane was, the molecular weight cut of the membrane was reduced by up to 38%. And as you can see from this this plot here, the removal of PFOS and PFOA were increased by up to 30% by the modified membrane. Now, we also tested how the other water matrix components interact, and we figured out that due to the coexistence of humic acids and cations like calcium in the water, the membrane shows up to 23% higher removal of PFOS and PFOA. Here we wanted to, explain the mechanisms of removals. Mainly it is because of the size exclusion as the membrane is being coated by this polyelectrolyte layers, and then also because of their complexation with the cations and the humic acids present in the water. We observed that higher concentration of cations would increase this complexation, and hence the removal of this PFAS by the modified membranes. Here is another example of membrane modification, but this time with the forward osmosis membrane. So we took a commercial forward osmosis membrane and coated with, again, polyelectrolytes, and within the polyelectrolyte layers now we embedded zero valent iron nanoparticles using this bench scale setup, and then we tested this membranes whether we are able to reclaim some water from impaired sources like stormwater or wastewater using this bench scale forward osmosis setup. As you can see on this same images, the membranes are, getting actually uniformly coated as you increase the number of layers. This is for 14 bilayers. Bilayer means one layer of the oppositely charged polyelectrolytes. This is the flux curve. As you can see, the unmodified membrane flux actually drops significantly, whereas the modified membranes, although they start with very little flux, but they maintain their flux throughout the duration of the filtration. And also they remove the nutrients very effectively. Here you see nitrate removal by the membrane. Oops, sorry. Now, this is an example of my research effort in modeling the fate or determining the fate of CECs in water. So this project is currently funded by the Fish and Wildlife Foundation of Florida. We are trying to determine the linkage of between the water quality in Volusia Blue Springs and the nutrient loading throughout the spring shed. But here we are also adding, we're investigating the effect of anthropogenic activities by including caffeine, investigating the fate of caffeine in the spring shed. So this is the boundary of the spring shed. So we've collected samples from the spring shed from various septic tanks and groundwater wells. And the samples are currently being analyzed for nutrients. So this is the partial result of some nutrient analysis, but I just this morning I received more results on nutrients, and we're now currently analyzing the samples for caffeine. I would like to briefly talk about two very recently funded or projects that are selected for funding. One of them is by Florida Sea Grant, where we try to control the pyrodinium, which is a major bloom-causing species. They will release the toxins, succintoxin, in their growth phase. So we'll here try to synthesize biochars from sargassum, which is sort of a seaweed, and try absorbing the pyrodinium and the succintoxins using the biochar. The other project also is funded by the DoD's Strategic Environmental Research and Development Program. Still haven't started the contract yet, so hopefully by the fall we'll start this project. But here, the PIs in Clarkson University will be testing a range of novel sorbents, to try to remove PFAS. But I'll work closely with University at Buffalo to test the efficiency of reduced graphene oxide and nano-zero-valent iron nano hybrids, their efficiency to remove PFAS or absorb PFAS as a function of various water quality parameters. So in brief, I'll continue my search for all-rounder membranes, and in addition, I'll investigate the removal of CECs and other contaminants using other treatment techniques. We have a project led by Dr. Naibin Chang from DEP, FDEP. So we're trying to remove PFAS and alkyl toxin and nutrients using bioabsorption media. In the long run, I plan to develop tools to quantify the interactions of CECs with membranes and water so that we can predict their removal and fate in the environmental media. With Dr. Lee, I have plans to develop membrane, smart membranes to be able to sense membrane fouling and other contaminants. And as an effort to model the transport of CECs, we're right now working on a proposal with collaborators from different departments to detect and model freshwater algae and algae toxin in the Lake River Estuary system in Florida. Well, I'd like to thank my lab, previous and current lab members. I'd like to acknowledge our department chair, Dr. Ati, and my collaborators and colleagues at CEC and other departments at UCF and outside UCF, and CEC office staff and ORC staff for their continued support. Thank you for your patience.